Welcome everyone. In this video, we'll discuss few genetics related problems, and such questions are very common in several of the competitive exams like CSIR NET, DBT JRF, GATE or the ICMR. Let's take few questions. We'll solve these questions one by one. Now, the first question is, in order to validate Mendel's experimental data, you make a cross involving parent pea plants of unknown genotype and phenotype. That means, the genotype and the phenotype is not known to you. And the following offsprings are obtained after the cross. You have the involvement of two genes, one for the pod shape, and the other one is the seed shape. You can easily figure it out if you have read the Mendelian's principle and postulates. Seed shape round wrinkled, round is dominant, wrinkled is recessive. Then you have the pod shape, inflated is dominant, constructed is recessive. What you have been asked is to determine the genotypes and phenotypes of the parent pea plants. So they are asking this unknown genotype and phenotype, and you have been asked to use the letter I for inflated and R for round. So, I mean, if you can look at these numbers, what we need to figure it out, how many of them are inflated. And if I can have a look at this ratio, the 3/8 is inflated here. And again, for the other phenotype, it is round. And in the second case also, you have 3/8 inflated. The total ratio, which in this population you are going to see, is 6/8 or 3/4. Inflated one gene, and then the recessive form of the same gene is constricted. And we can easily figure it out. The ratio of the constricted is 1/8 and 1/8, which is 2/8 equals to 1/4. If you can closely see these numbers, what you will soon realize that these are nothing but the ratio for the Mendel's monohybrid cross. So this was the same ratio which was obtained 3:1 for the monohybrid cross when the F1 species were selfed. What does it tell you? It tells me that the inflated gene is in a heterozygous form. The inflated gene is in a heterozygous form, which I can easily represent capital I, small I for one parent, and then the capital I n, small I for the other parent. Now let's talk about the other gene, gene 1 and gene. Again, we need to talk about the other gene too, because this is a dihybrid cross. So now let's have a look at the ratio of round and wrinkled. So the ratio of plants having round seeds are 3/8 plus 1/8, which is nothing but the 4/8, and which eventually will become 1/2. How about the wrinkled one? So if you see for the wrinkled one too, we have 3/8 coming from here and then 1/8 coming from here, which again in this case is going to be 1/2. Now what does it tell you? It tells me that for pod shape, or rather for the seed shape, sorry, the round and wrinkle is for the seed shape of the pea plants, and the gene 1, which is for the pod shape, dominant and recessive, inflated and constricted. Now, if you look at this ratio carefully, you will soon realize that the genotype for the seed shape is which I have to represent in the form of capital R and the small r. One of the parent has to be heterozygous, capital R, small r, and the other has to be a small r and a small r. So what I'm trying to tell you is I need to erase it, or let me try here. You have, the genotype and the phenotype for the parent is going to be, for one gene we have already seen capital I, capital I, small I, and then for the other gene, one of the parent is heterozygous for the round ones, or heterozygous for the seed shape, and the other parent is homozygous recessive, which in this case is going to be small r, small r. Now let's have a look. Is you can confirm it. What I'm trying to tell you is if you cross the parents of such kind of a phenotype and genotype, by the way, what is the phenotype going to be? This is going to be inflated because of the capital I and round, and this one is going to be inflated and a small r is for the inflated and round, sorry, one parent, and the other one is the inflated and wrinkled. Now let's confirm one of these ratio by making a cross by using such kind of a genotype. So what we can do is, let's have a look, is if I'm making a cross between such kind of a genotype, let me clean the slide and rewrite it again, is I'm making a cross between capital I, capital R, small r, and the other parent is capital I, small I, again heterozygous, and the homozygous recessive for the other gene, small r, and let's confirm whether I'll get 1/8 constructed wrinkled or not. Now, if we remember, for the constructed ones, the genotype has to be small I, and if I mean, it's a simple thing, is either you can go ahead with the Punnett square, which will take much more time, or you can just make use of the probability. So by considering one character at a time. So if I'm making a cross between capital I, small I, and the capital I and small I, the probability of getting the plants having a constructed pod shape is 1/4. And the reason being, again, the Mendel's postulates, law of segregation, law of independent assortment, and you'll get 1/4 constructed. Fine. And how about the wrinkled ones? If I'm making a cross between capital R, and a small r and a small r, I'll get capital R, small capital R, small r, and then a small r, and then a small r and a small r. Fine. So 1/2 is, you have the 1/4 and 1/4, which is nothing but the 2/4 or 1/2. Now, law of multiplication, and the total ratio for constructed and wrinkled is going to be 1/8, and that's what I'm getting. You can confirm it for the other ones and you will get it. Now what you need to understand is, you need to think is, at what point I need to keep the genotype in a homozygous condition or in a heterozygous condition. And even if you are randomly putting any of these genotypes, you need to confirm those by making a cross. So you'll get it. So coming back to this, and coming back to the answer of this question, is the parental genotype is capital I, capital R, small r for one parent, maybe for the male pea plant, and the female pea plant is going to be capital I, and the small r, or even the vice versa. I'm just considering one at a time. And the phenotype is going to be, as said, inflated and round because of the presence of capital R, and then inflated and constricted. I hope this answer is clear to each and every one of you. Now let's move to the second question. So suppose that you are doing a tri-hybrid cross between two plants having a given genotype, and the genotype is given. It's a heterozygous for three genes, very clear. There is an involvement of three genes, EF, and G, the capital ones are the dominant, needless to say, following the Mendelian principles. And what it has been asked is what fraction of the progeny or offspring will show the recessive phenotype for at least one of the given gene. Now, read it very carefully. What fraction of the offspring will show the recessive phenotype for at least one of the given gene? Now, there are many ways you can solve this question, but remember, if such kind of a questions comes in the competitive exam, you need to be smart enough to solve it in a very minimal amount of a time. And what I would recommend you is first we'll go with a the way where you need to think and you can solve it in a minimal time. And then again later on I will show you the second one where you can take your time and you will get the same answer. So if I have to say so, what fraction of the offspring will show the recessive phenotype of for at least one of the given gene? So for at least one of the given gene, it could be even three, it could be even three, it could be even more than one, but at least it should be one. Now remember, I can reframe this question for the solution in some other way. For example, I can rewrite is what fraction of the progeny or offspring will not show the recessive phenotype for even one gene. And what I'm trying to tell you is, I mean, this is the situation. For example, E dash F dash and G dash. Needless to say, these will form 8 gametes and there are going to be different offsprings of the different genotype. And what you need to figure it out is the offsprings which are carrying a recessive phenotype for at least one of the given genes. So for example, you can think of any of the offsprings which having a genotype small e, capital F dash G and dash. Dash means it could be the small g or the capital G. Doesn't matter because if the F and G are the dominant ones, even a single copy is sufficient enough to show its expression. Or the second could be small e, small of small f, and then capital G dash, or even the third case is small e, small f, small g. You can keep on writing. That is going to take time. If I'm correct, those are going to be total 7 in number. But what I'm telling you is you can solve the same question in a different manner. So what I'm asking you to do is you just read this question in a different manner. You can think of what fraction of the offspring will not show the recessive phenotype for any of the gene, and the answer is going to be if our offspring is going to have the genotype something like this, is you have a single copy for each of the gene. Now what you need to understand is what is the probability for an offspring to carry this kind of a genotype. So as discussed in the class, as I've said so many times, is what you can do is for such kind of a probability calculation, you can consider one gene at a time. So what I'm asking you to do is you consider capital E small e and then you cross it with the capital E small e because the genotype of the other parent is the same. And then you just try to figure it out what will be the probability or the fraction of the offsprings which are going to be carry, capital E, small e or capital E and capital E. And what you will find, this value is going to be 3 by 4. You just figure it out, is 1 by capital E small e from one parent, capital E small e from another parent. So you are going to have capital E, capital E, capital E small e, capital E small e, small e, small e. And what I'm asking is, are these three ones, which are nothing but the 3 by 4. Same for the F, 3 by 4, and same for the G, which is going to be 3 by 4. And what you need to apply here is the law of multiplication, because I'm asking for a offspring which are having a genotype capital E dash, which could be anything small e or the capital E and capital F and dash, and then capital G dash. So though that number is going to be 327 by 64. Correct? So this is the proportion, this is the number, where you'll find, this fraction of offsprings which are not carrying even a single copy of a recessive gene. But this is not the question, which has been asked. What has been asked is what fraction of the progeny or offspring will show the recessive phenotype for at least one of the given genes. So what I need to do is, remember, the probability lies between 0 to 1. And what I can do is I can subtract this value from the 1. So 1 by 27 by 64, and the answer which you will get it is 64 minus 27, which is going to be 37 by 64 if this calculation is correct. Yeah. So the correct answer is going to be 37 by 64, because what they have asked is the fraction. You can report it in a fraction, or you can even convert it into the percentage depending upon what has been asked. So this is the simpler one. What you need to consider is, I mean, there are going to be many possibilities for the offsprings to carry at least one of the gene, which is, which will show the recessive phenotype for at least one of the gene. But what I'm asking you to do is, you just calculate the number of offsprings which will not carry a single recessive gene for capital E, capital F, and capital G, and that number is going to be 27 by 64. You subtract this value from 1 and you will get 37 by 64 as a correct answer. So the correct answer is going to be, sorry, 1 minus 27 by 64, which is going to be 37 by 64 in terms of fraction. So these are the offsprings. Those are going to show the recessive phenotype for at least one of the given gene. Now let's take a longer route. This is what I was talking about. What has been asked in the question is the recessive genotype for at least one of the gene, and what are the different possibilities of the genotype is small e here, again small e, small f here. Remember, they have asked at least one, could be more than one too. Again, where all the three are recessive, here one, two genes are recessive, and then again one, two genes are recessive, correct? And you just try to calculate the ratio for each of these genotypes. And these are the ratio which you are going to find. Needless to say, what you need to consider for calculating these ratio is, you can simplify your calculation by considering one character at a time. So what I'm trying to tell you is capital E, small e, capital F, small f, capital G, small g, capital E, small e, capital F, small f, capital G, small g. This is what has been given in the question, and what I'm asking you to do. For example, let's take one is, like, if I'm making such kind of a cross, what is the fraction of the offspring which I'm going to get for the given genotype, capital E blank, which could be a small e, or the capital E, a small f, a small f, small g, a small g. You just make one cross at a time. So if I'm going to cross capital G, small g with the capital G, small g, this value is going to be 1 by 4. This again is going to be 1 by 4. How about this one? This one is going to be 3 by 4. And what you need to do is the law of multiplication, and the value which you'll get is 3 by 4, 4, 16, 64. And this is what I have reported here. Remember, you can do it this way. This is going to be much more tedious, but you'll get the same answer. Finally, at the end, what you need to do is you need to add everything, because what has been asked in the question is, what fraction of the progeny will show the recessive phenotype for at least one of the given genes. So what I need to apply here is the law of addition. These are the possible ones. Could be this one, or this one, or this one, or this one. And if we'll add up all these 7, we'll get 9, 3, 12, 12, 13, then again 21, 24, 27. Have I made any mistake? 9, 9, 18, 27, 30, 33, 34, and 37 by 64. That's what we have calculated, is we'll get a fraction of 37 by 64. I hope this is clear to each and every one of you. Let's get back to the third question. Again, this is a question from the genetics, and what it has been asked is, you need to perform the statistical analysis. So let's read this question carefully. In certain species of mouse, hypothetical situation, black fur color is dominant to albino. So what does that mean is black be dominant to, or rather, yeah, albino. And then you have the rough coat, which is dominant to a smooth coat, a small r, a group of heterozygous black smooth males. So what you need to do is, from this statement, you need to figure out the genotype, a group of heterozygous black. So for the black, it's capital B, small b, smooth males. And it's for a species to be smooth, it has to carry a homozygous recessive condition, which is small r, are mated with a heterozygous black, again capital B, small b. We all know what is heterozygous is both the two variants of the gene are present, or rather the two alleles of the same gene are present, capital B and the small b, and those are not identical. All right, mated with heterozygous black, heterozygous rough females, and I need to represent the rough with capital R. Remember, it's given heterozygous rough females, so it's a capital R, small r. And then, the given number of offspring with the following phenotype are produced. Remember, what the values which are given is the observed. And what you need to do is you also need to calculate the expected ones. And then you need to figure it out. Like whatever differences you are seeing in the observed is just because of a random chance, or there is a statistical significance. And based upon that, you need to formulate your null hypothesis. You need to calculate the chi-square value. And then it has been asked whether you accept or reject the null hypothesis. We all know what is null hypothesis is we can create a null hypothesis based upon whatever information is given there in the question, and the hypothesis which can be created is we can consider the black fur, which the capital B is dominant to albino, and the rough coat is dominant to smooth coat. So we can say that the coat morphology is dictated by a rough one, a dominant rough one, and a smooth one, recessive smooth one. Same. The fur color in a certain species of mice is dependent upon the dominant black fur, which is represented by the capital B, or the albino one, which I can represent it with the small b. So you can even say that, the fur color in mice has two phenotype, which is black and albino. I mean, you can create any of these hypotheses. And the rough coat in such kind of a the in such kind of a mice, not rough coat, the coat morphology is the rough and smooth, and the fur color is the black and albino. So you can create this hypothesis. The fur color is the black and the albino, and the coat morphology is the coat appearance is the rough and smooth. Or you can say that, additionally, you can say that the black fur color is dominant to albino, as well as the rough coat color is dominant to smooth. Now we need to, like, we have created the hypothesis, we need to prove it. And how are we going to do that? With the help of the statistical test, which is a chi-square test. Now the hypothesis is done. Mention the calculated chi-square. If you look at the formula, this is how we calculate. Observed, given, expected, I need to calculate, which I'm going to show you in a moment. And then you would need to do observed minus expected chi-square divided by expected, and summation of all these values will give you the chi-square. Now let's have a look. We have already figured it out, the genotype, in the previous slide. This is what I was talking about, is when I'm making such kind of a cross, what are the expected ratios? Now when I'm saying expected ratios is, let's have a look, is if I'm making, for example, if I'm saying, so black and rough, what is the genotype going to be, by the way? Is, remember, black is dominant, so it could be B dash, and rough is capital R dash. And then again, black and smooth is going to be capital B, and smooth. I'm just taking the letters from here. For a smooth appearance, you need to have homozygous condition smaller, smaller. How about the albino? Albino is going to be small b, and albino and rough again. R and sorry, R and dash. There is no dash here, by the way. And then you have the albino and smooth, which is, both the two are recessive. So let's have a look. Calculate these ratio. For example, let's calculate for one of them is, if I'm making such kind of a cross, what is the probability of getting B dash, R and dash? So for example, if I'm making B, you can just the same thing I said few minutes back, you can take a single character at a time, capital B small b with the capital B small b, and what is the probability of getting capital B dash? Nothing but a 3/4. Same for the capital R, small r, and then small r and small r. So what I need to get is the capital R and S, and those values are going to be capital R, small r, capital R, small r, sorry, small r, small r, small r, small r. So this again is going to be 1/2. So this value is going to be 3/8. So what I'm trying to tell you is the expected ones. That means if I'm making a such kind of a cross, I'll get a ratio of 3/8, which are going to be black and rough. Same you need to calculate it for the black, smooth, albino, rough, albino and smooth. And I have calculated it for each of these phenotypes. So we'll get 3/8 for the black, rough, 3/8 for the black, smooth, albino, rough, 1/8, and same for the albino, smooth. Now remember, observed is already given. Observed is already given in the question, these numbers. What I need to do is I need to calculate the expected ones. If you'll add all these species, you'll get 150. Now what I need to do is, if I'm saying 3/8 fraction of what? 150, which is going to be 56.25. Again, what you need to do is you need to be smart enough to round it to the nearest whole number. So I have considered 56 in both the two cases and 1919 because the first number coming after the decimal is more than the 5. I mean, I hope this is clear to each and every one of you. Now, if you can go back and see this formula, observed done, expected done. I'm aware with the expected. What I need to do are these simple calculations, and that's what is shown here. You have observed given, then the expected, which we all have calculated, and then the observed minus expected difference. Remember, 4, you will get 4 here, -3, -4, 3. All these will get converted into positive because, the next step is the square. So 416, 916, and 9. And then you divide it by the expected one, observe, and you will get these numbers. Now, for calculating the chi-square, what you need to do is you need to add up all these numbers, and what you will get is 1.762. This is the same thing. That's what they have asked. Is the mention the calculated chi-square value, which is 1.762. It's always advisable you consider, till 3 digits so that your chi-square value is much more closer and accurate. Now, the next thing we need to understand, we need to have a look at the probability table. Is whatever differences which I am getting again is just because of a random chance or a chance event, or is there any statistical significance? And that I need to compare by looking at a table. And remember, if I have to go through that table, I need to calculate the degree of freedom. And what does that mean? It's a simple total number of categories given in this question. Those are nothing but the 4 ones. These are the 4 ones. So if you look at the formula of degree of freedom, it's n-1, where n represents the total number of categories given, which is going to be 4, and then 1 more. You subtract 1 from it, which is going to be 3. So we have the degree of freedom 3, we have the chi-square value 1.762, and then I need to have a look at this table. So I mean, what is given on the vertical axis is the degrees of freedom and what I need to focus here. And then I need to figure it out, where is my chi-square value lying? It's lying somewhere in between these two values. And remember, it's much more closer to this value. And corresponding to that, I need to find out the p value, which is nothing but a probability value. For any of the statistical significance, we consider, the p value of 0.05, but what we can actually see here is, the probability which is coming here for our question is close to 0.50, which is nothing but a 50%. What does that mean? It tells me that there is a 50% probability, there is a whatever difference which I'm seeing between the observed and the expected ones, it's just because of the 50% chance event. So I mean, the differences are merely because of the chances. And what I need to do is, I don't need to create a alternative hypothesis. I just need to accept the null hypothesis. Now remember, what if you are getting values like chi-square values like 8 or more than 7.815 or 7.815? In that case, your p-value is going to be just 0.05, which is nothing but a 5%. Remember, what does that mean? That tells you that the differences which you are seeing between the observed and expected is only because of the 5% chance event. So there is only 5% chance event. So what you need to do is you need to reject your null hypothesis. That's what they have written here. And you need to come up with an alternative hypothesis, which could be anything. If I have to create, let's say, the alternative hypothesis, I can say that is either the black fur color is not dominant, or uh, the fur color in the mice is not only dictated by the two genes. There might be a role of a third gene, or could be there is a presence of the environmental factors or some kind of a food and all. So I need to again create some alternative hypothesis and I need to prove it. So I mean, creating an alternative hypothesis doesn't tell me that whatever I am creating is correct. Again, I need to prove it with such kind of a statistical calculations. So going back to the same question which was asked, is mention the calculated chi-square value, which we have calculated 1.762 if I am correct. Yeah. And then do you reject your or accept your null hypothesis? We accept, the reason being the p-value is coming to be 50%, and there is a, what does that tell you is that there is a 50% probability of the chance event between the differences in the observed and the expected value. Explain your answer. This is all you can write it out, and we all know, unless until only if the p-value is coming either 0.05 or 5% or less than 5%, then only we reject the null hypothesis. Include in your answer p-value, you can write the range, or even if you're writing, the 0.5, it should be okay. All right. I hope this is clear to each and every one of you. Now let's move to the last question. Is what is given here is a couple both heterozygous for cystic fibrosis have 5 children. So this is what is given is a couple both heterozygous for cystic fibrosis have 5 children. So already mentioned that they have the 5 children, the couple. What is the probability that at least one child will be suffering from cystic fibrosis? Now remember, it's an autosomal recessive disorder. What does that mean? Is it's not, of course, going to affect the sex chromosome. So it's it is on the somatic cells or the autosomal cells. So nothing to do with the boys and girls. These are going to affect the children irrespective of their sex. So autosomal recessive disorder, so let's represent it, is a couple both heterozygous for cystic fibrosis. So let's represent the cystic fibrosis with the A. So both heterozygous for the cystic fibrosis. So we can consider capital A small a and then the capital A small a. Now there is a cross between such kind of a genotype, and what it has been asked is what is the probability that at least one child will be suffering from cystic fibrosis. So they have 5 children, 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. And what is the probability that at least one child will be suffering from cystic fibrosis? Now the same thing, which I did it few minutes back. You can reframe the question. What is the probability that none of the child will be suffering from the cystic fibrosis? And in that case, each of these offspring has to carry a genotype of capital A dash, capital A dash, capital A dash, capital A dash, capital A dash. Because remember, it's autosomal recessive. If an individual has to suffer from this disease, if a child is carrying this disease, the genotype is going to be small a, small a. And what they have asked is at least one child. I mean, at least one child could be more than one. It can be, it is possible that even all those five might be carrying those cystic fibrosis genotype or the disease. Now, what I'm asking you to do again, I mean, to make the thing simpler, you can just think of what is the probability that none of the child will be suffering from the cystic fibrosis. And the answer is going to be is if you are making such kind of a crosses, capital A, small a, capital A, small a, what is the probability of getting capital A dash? So that means capital A, capital A, capital A, small a. This is what we will get, capital A, small a, small a, small a. Remember, it's a 1/2 law of segregation. So these two will segregate and will form the sex cells, haploid sex cells. Remember the meiosis. Even the same thing will happen here. So you have the 1/2 probability, 1/2, and you have, these are not going to be suffering from the cystic fibrosis. And that's what I have asked you to consider to simplify this calculation is what is the probability that none of the child will be suffering from cystic fibrosis in such a condition. And the answer is going to be for each of these is 3/4. Remember, 1/4, 1/4, 1/4, and you just need to add those three. 3/4 for each of these child it is going to be 3/4, which is nothing but it's a law of multiplication. Remember, 1, 2, 3, 4. I think one more is left. 1, 2, 3, 4. Sorry, 1, 2, 3, 4, 5. I need. You can even write in such a fashion, which is going to be 243. Let me use my calculator. So this is going to be 243 divided by 1024, and the value which you will get is 0.237. But remember, this is not the thing which has been asked. It is asked what is the probability that at least one child will be suffering from cystic fibrosis. So this is the probability for a situation where none of the child will be suffering, either 0.237 or you can convert it into a percentage close to 23.7. Now what I'll do, as we all know, the probability lies between 0 to 1. So 1 minus 0.237, which is accurately will give you 0.763, 0.763, which is nothing but a 76.3%. So what I'm trying to tell you is what is the probability that at least one child will be suffering from cystic fibrosis is 0.763 or 76.3%. You rather mention in a form of 0.763, which makes much more sense, and not in a form of a percentage. So what does that mean is there is a 0.763 probability which will make sure that at least one child is suffering from the cystic fibrosis. So there is a much more higher probability. I hope these solutions or the explanations are clear to each and every one of you. If not, you can write your comment or feedback in the comment section. And even if you have any additional question, you can put your question there. I will solve it in the next video. All right, thank you very much.